This is going to be our lab report for Lab 1. It's the original one we had with the dried peas and the beets and the seeds. And our group members are myself, Andray, Ilan, Kathleen, Adam, Jonathon, and Brenda. And the basic background to this lab is stated here. I'll read it for you. This lab focuses specifically on aerobic cellular respiration that takes place in plants that do not acquire energy through photosynthesis due to being underground. The reactions that occur during aerobic cellular respiration are divided into 4 different steps: glycolysis, acetyl-CoA, synthesis, Krebs cycle, and electron transport chain. The primary purpose of this experiment is to it's going to be comparing the respiratory rates of 2 different types of peas that we use throughout respirometers. So basically we're learning about respiration, and we're comparing 2 different types of samples to see which respires more. Okay, these are going to be our lab questions, and this was done by, I believe it was Kathleen. Anyways, our hypothesis is how fast do different kinds of beans respirate? So that's going to be the overall question that we're trying to answer throughout this experiment, because we have 2 samples and we're going to be comparing the respiratory rates of each one. So as you can see in our graph, times on the x-axis, volumes on the y-axis, and due to respiration, volume is going to be displaced, and it's going to be decreasing over time and at a semi-steady rate. So that's this graph is a depiction of that. On this right-hand side, you can see other basic simple questions are being answered. For example, this one, we're supposed to be comparing Table 1 and Table 2. And from this, we can infer that the dried peas didn't undergo as much respiration as our other peas, and that's going to be respirometer 2, so it's going to be remaining constant for most of the time. Also here, we just plug in some numbers to this formula, y-axis over the x-axis, and that determines the rate of oxygen consumed for each treatment. These are our pre-lab questions, and these were also done by Kathleen. The most important part of this section is basically what variables do you think affect respiration rate of seeds? Brainstorm within your group, generate a list. And we came to the conclusion that basically the amount of seeds and the amount of beads in comparison are they heavily influence the results of our respirometers, and also the time spent in the water. So as time went on, respiration can either be stagnant or it will be increasing little by little, as was shown in respirometer number 1 with the germinated peas. Okay, up next is our lab procedure, and this was done by, I believe, Brenda. And basically this is the steps that we're going to be going through in our labs, as was stated in the packet. So this is just basically overall outline step by step of each thing we did. So I'm not going to go ahead and read through all of this because it's a lot, but yeah, this is our lab procedure. This is our data analysis portion of our lab report, and basically all the results that we took throughout the report through observations and experimenting are going to be summed up neatly on this chart. I'll start over here with respirometer 3. That was beads only, and that was meant to be a control. So basically everything was stagnant. Respirometer 2 shows dry peas and beads, and the beads were only there to match the volume of the peas in respirometer 1 because the dry peas were smaller. So what we were shown in respirometer 2, however, was that they were not respirating because there was no change throughout the whole time. Respirometer 1 gave us the best results because, as you can see, the volume of the pipette decreased each time as we took measurements. And our data analysis, we can basically conclude that the germinating peas showed a steady rate of respiration, while the dried peas showed no sign of respiration. This is going to be our hypothesis restated again, in case you forgot. This is an alternate wording of Kathleen's hypothesis early on. Will the results shown in respirometer 2, dried peas and beets, have a lower rate of respiration than germinating peas? So basically we're trying to find out if there's a difference in respiration between the 2 samples. And our conclusion at the end of the experiment was the germinating peas did not end up having a higher rate of respiration than the dried peas. This was expected due to the dried peas being basically dead, ceasing respiration. And that's all. Thank you.